All right, well, we have time for a chat now. This is sort of panel discussion time. So if anyone's got anything that they think, I mean, it's been a fantastic variety of talks today. If there's anything that has really sort of drummed up a thought in someone, a question that maybe we need to explore further, it is a wide range of topics there, but is there something that anyone would like to discuss? It's the end of the day, isn't it? We're all switched off. Oh, well, I guess I'll throw something out there then. So one thing that we talked about is probably not relevant to all the talks today, but certainly with a lot of them prior to the break as well. We talked a lot about pests in different crops and, you know, all these different ways to monitor and manage them. But I guess one thing, and it certainly crops up at CSER Australia, where I work, is how do we implement this? So, you know, we're doing all this fantastic research as scientists, but really how do we get it out there, providing the growers, providing the general public with these tools? Has anyone got really any ideas or you know, are they aware of any shortfalls in terms of the extension side of this research in this space? Oh, and whilst you're thinking about that, we have another question. So Francesco, are those surveillance trailers being used Australia-wide, and by who, if so?
Yes, they are currently. So it's obviously, you know, an R&D program. So since we started in 2018-17, started building them, the things were, you know, a steep learning curve. So we started, the first two are actually trailers, and then from there on they realized that that was actually too much energy consumed. So they are actually put them on top now of pickups, so they are smaller like box, as Kevin is suggesting. So we have used them in Meringa. We have one currently here in Victoria, and they are a bit everywhere. So yeah, so they are used. We have been using them on many different kind of crops. So from sugar, currently are sugar, cotton, ongoing in grains. We are starting tomorrow in Mildura for this citrus sunraysia test as well. Obviously, you know, again, research and development. So we were trying very different areas, trying to see if different environments were sort of impacting a bit the things. So we had a few of them breaking down mid-trial. So you know, what you would expect from something that you are testing. Yes, so as Kelvin is suggesting, obviously one of the major things is also trying to compare sentinels with normal, more traditional ways of collections. So for example, in the trial that will start tomorrow, we have both glycol traps around the sentinels. Then we will put some glycol traps afar, just see if the distance from the sentinel can be a factor. Use other suction traps as well, wind traps with David Medge. We have them, prepared. So there are, you know, obviously a lot of questions out there. Even the first 2 or 3 sentinels actually have 2 towers, a 6-meter tall one and a 2-meter tall one. And guess what? You will find a difference in the catch, even just so same sentinel, but 2 different towers. We are collecting also fungal spores and testing for bacteria as well. I mean, I don't think that that's obviously the only way to collect. You will always need to pair them, and most importantly, metabarcoding is not the only way to analyze the data. One of the issues, answering Samantha's question, is probably that obviously, you know, to have data that is meaningful, it needs to be very, very, very fast. You can't, you know, take 2 weeks to analyze the stuff, or just wait, you know, a full week of collection and then have the strap sample shipped on the other side of Australia, do the sequencing. That's, the data is not useful anymore. So you obviously have to differentiate if it's something that needs to be targeted and known immediately because farmers need to spray the field as soon as they find the first specimen, then that obviously needs to be done following a different path. But yeah, I think obviously that remains one of the major issues. Yeah, we still need different ways to tackle different issues. So yeah, tailored approaches. Yeah.
Thanks for that, Francesco. Have we got any more questions for any of the speakers? Helen, there's a question in the chat for you. Did your work note any trends in first records of pests? For example, surveillance versus research projects versus industry versus citizens?
Yeah, okay. So we only had that for the last 35 things. That there haven't been very good records of who was making detections. So that was, I think that went back to the late 80s. And no, not really. So we looked at whether detections were being, like, who was making them and where they were being made in terms of urban areas and regional areas and nurseries and blah, blah, blah. And there weren't really any, just that surveillance wasn't picking up as many things as possible. So I don't know if you know, forestry is about to roll out a nationally coordinated surveillance strategy, and one of the things is to try to make detections earlier so that eradication is more feasible. We've actually only ever had one successful eradication in forestry, not including the giant pine skull was successfully eradicated from Adelaide, but it's still in Victoria, so we can't really count that as it was successful in South Australia. But we've only had one successful eradication of a forestry pest so far. And part of the reason for that is because things aren't being detected soon enough. So the whole idea of the national program is to try to be able to detect things early enough for eradication to be successful. So that's why we were looking at who's making those detections, just to try to work out how we can make that a little bit better. But the data set was too small to really see any good trends over time for that. But I think public have been pretty consistent, and with things with like with iNaturalist and Quest a Game and stuff, I think that's only really we're going to have more, like, so our list stopped with 147. We've had a few more things since then, and they've been public detections as well. So go public eyeballs. I'm quite a fan of it. I know there's a lot of processing of it because you get a lot of things that aren't anything, but it just seems so much more efficient. So many more people can be looking. Yeah.
There's another question for you,
Yes, it's New South Wales. And another interesting pattern that we found was a difference in the types of things that are getting in. So Queensland is receiving a lot more things from Asia and Pacific, which makes sense with the geographic proximity and climatic similarity. But a lot of things are getting in or being found in New South Wales, at least, and moving from there. So actually the source of most invasive insects to any state is other Australian states as well. So that was interesting. There's so much in what we've done. It's just crazy, really. Yeah. But cool.
Thanks for that, Helen. Any more questions? We've probably got time for a couple more. Or any comments that anyone's got to make on any of the talks?
I'm really excited about metabarcoding, Francesco, as we do a lot of trapping in forestry, and yeah, sorting through hundreds and hundreds and hundreds and hundreds and hundreds of bark beetles. Oh my God, they all look the same as it is. I hate them. So being able to meta barcode trap catches for forestry, I think will be fantastic. We are part of the IMAP Pest project, but not a huge investor. Like I think forestry had a bit of FOMO with that. It was like, no, we want to do it too. So we haven't. I think they've done a couple of Sentinel stuff, but a lot of the things that we're looking at aren't really amenable to those suction traps. For a lot of our pests that are on the lists and stuff, we have no Hemiptera on our high priority pest list.
I know. So we actually have tested some forestry traps, but not from the Sentinel. So they were sent to us, and man, they were dirty, like super dirty traps. So that's really
dirty
forestry, because then obviously, you know, with a lot of pine needles and everything in it, quite some issues there with PCR inhibitors. So we really have to find like a way to have traps, I don't know, more clean or collected more often. But then obviously that, again, it's a trade-off between, you know, having someone that has to go out, so time expensive or just expensive, but yeah, it's very interesting. Like the last time I actually tried with some of those traps, they were so dirty. I ended up unfortunately finding 2 frogs in them that I couldn't even see from outside.
I know.
So really dirty.
Yeah, I know. We get geckos sometimes.
That stuff is, you know.
Bats. Yeah, I know.
Francesca, we've got one more question in there as well, which will probably make the last one. What proportion, this is from Luke, what proportion of barcodes in meta barcoding do not have suitable barcode matches in the databases?
So yes, that's obviously one of the major issues. Not if I had to tell you a proportion, I don't know, probably we end up being 20%, including in that both stuff that matches sequences on public databases, but the sequences are not identified to species. So for example, you have 100% match, but the species is just called Acacia sp, and that's not really helpful. I mean, it gives you information on where else that same species has been recorded, but it's not a species level identification. And then you have obviously something that doesn't match absolutely anything, and the closest match is 80%. So you start doubting yourself, is this an actual insect or not? But that is something that is obviously improving a lot in time. So when we do have very interesting specimens, one of the strong point of doing the non-destructive DNA extraction is that you can go back to the trap and re-extract DNA just from that specimen, and then maybe have a full-length barcode, full cytochrome oxidase. I mean, you could even do the whole mitogenome if you wanted. So it's something that it's improving with time, and it shows the huge importance of having a curated database, because the second big issue there is that you obviously do all this through a pipeline, a bioinformatic pipeline. You have so many reads. We're talking in the millions, so millions reads of DNA for every run. So you obviously cannot do that manually, which means you need an algorithm that goes and automatically blasts it and takes the best result. And that unfortunately is biased by wrong taxonomical assignments on public databases. So even if you have 99 sequences that are correctly assigned, but you have another one that someone has misidentified and uploaded on GenBank, your algorithm might not know which one of the two is the correct one, and it doesn't assign it to species. So that's really painful. So what we are trying to do is moving out from public databases. We use those sequences, but we curate the database, and then we use our curated database to do the analysis. So yeah, a big issue, yeah.
Thanks so much for that, Francesco. Well, we're dead on time. Oh, minute over now. So I just want to say, if everyone can join me in thanking all the fantastic speakers for today. So thanks for that. It was really, fascinating talks. Now, I think the next stage is the IUSSI AGM is on now. So, if that's your cup of tea, see you there. But otherwise, I think tomorrow morning, Andy Austin's giving a talk at 11:30 Melbourne time. So thanks, everyone, and have a lovely evening, and catch you soon.
Thank you, Samantha. Bye-bye.
Bye.
Thanks, Sam.